Right, everyone's coming in. All right. Hello, everyone, and welcome to today's webinar, A Fundamental Guide to Reprocessing Medical and Other Devices. Thank you for taking the time to be here today. I'm Matt, and I'm the Senior Marketing and Events Specialist at SSS Australia. We need to inform you that this session will be recorded for future training purposes. We would like to thank our speaker today, Dr. Whiteley, who is the Executive Chairman of Whiteley Medical, a manufacturing company that develops and manufactures medical chemistries for medical device, reprocessing, and healthcare surface cleaning and disinfection. Dr. Whiteley has expertise in the measurement of surface hygiene and supervises an extensive collaborative research portfolio into medical biofilms and biofilm-mediated infections. Dr. Whiteley is also an adjunct associate professor in the Faculty of Medicine and Health at the University of Sydney. You'll be able to find out more about Whiteley Medical and related products in our follow-up email, which will go out after the webinar. If you have any questions, please click on the chat box located at the top of the screen. These will be monitored during the webinar and relevant questions will be answered at the end if we have time. If we don't get to them all today, we will get responses out to you next week. Keep an eye on the chat screen as well when we post a link to the feedback form. Here you can let us know how the learning session went, and through this form you can request an attendance certificate. A link to the video of the presentation will also be sent out next week. Welcome to anyone who has just joined. I will now hand over to Dr. Whiteley.
Well, thank you very much, Matt, and thank you all. I'm just going to share screen and we'll go to share screen. There we go. Has that come up, Matt?
Yes, looking good.
All right. Well, welcome everyone, and thank you for your time this afternoon. You can ask questions in the chat box as we go along, but I will answer questions at the end. There are quite a lot of slides that we'll be going through today, and I want to encourage you that you don't need to take huge notes because we will make these slides available afterwards through SSS. I'd like to thank the SSS team also for the opportunity to talk. A little bit more background just on myself. I formerly or previously served on HE023, which is the Standards Australia Committee that is responsible for this group of standards. Our representative on that committee now is Dr. Trevor Glasby, and I'd like to give Trevor particular thanks in the preparation of these slides. Trevor also not only sits on HE023, but he sits on the Superior International Standards Organisation Committee of TC 198. And represents Australia there with some other Australians. The other thing to note for me is that I also serve as a consultant to the Australian Dental Association's National Infection Control Committee. So this is an area we know well. It's an area that is of great interest, I'm sure, to everyone. And I'm going to click and hopefully that will go down. So just a bit of a outline of what we're going to do. We're going to do a bit of a history of the standards because that's highly relevant. There's been a lot of emotion about this standard in some sectors. We're going to talk about the importance of cleaning and infection prevention. We're going to mention the Spaulding's classification scheme, a little bit of look at the old standards and the implications of the new standards, the big impacts, and then questions. And as I say, if you have questions, please pop them in the chat box. Now, I joined HE023 myself back in literally the 90s and have been involved for a long time. We've been intimately involved with these standards. Initially, the precursor standard was NZS 4187, and it was initially released as a code of practice. It wasn't even a standard and developed by Standards Committee HE002. And that was then peeled off into a much broader standard. And when that went out into the field, there were a whole pile of issues that turned up that really needed almost immediate adjustment. One was in our particular area that in 1996, the Therapeutic Goods Administration gazetted a whole pile of new guidelines that weren't fully reflected in the early edition of 4187. And also, there were some real issues around the implications for office-based practices. So let's just look at this little schematic. So initially in 94, we had what was 4187 as a code of practice. It moved to a standard. Almost immediately it was realised that there was a bit of an overreach, and particularly the office-based practices were being dragged into what they perceived as a huge amount of bureaucratisation. And the Royal Australian College of General Practitioners and the Australian Dental Association, with the Australian Dental Industry Association and some others, were integral in making sure that we split apart the standard so that it really became clear what was the implications for office-based practices versus the large teaching hospitals and large private hospital groups. One of the immediate things that became obvious was that the office-based practices generally were not doing overly complex procedures. You know, they're not doing orthopedic hip transplants and so forth, or replacements. Whereas, of course, in the big primary care facilities, there were obviously a full array from simple things to complex procedures. And as we move through the time, and you can see from this that we had revisions out in 2001, 2003, 2006, 2014. The irony of the 2014 standard was that it was to take effect from the 30th of December 2023. And ironically, the new standard 5369 was published before the old standard of 4187, or the most recent revision, became active. But that 5369 now brings together all of the standards under a single envelope. And if you look at what they used to do, as I said, 4187 was really about the big healthcare facilities who had lots of staff, and with more staff becomes more complex training needs, more complex supervision, more complex record taking. Whereas you go across into the 4815 area, office-based facilities, GP clinics, but it also picked up places like beauty parlours, tattoo parlours, and an array of other applications where sterilisation was relevant. And there was a whole pile of issues about what they could and couldn't do. It was a much less regulated environment. And then over the top of all of these standards, particularly from 4187, the most recent revision, there were a whole pile of superior standards, particularly under the group of ISO 15883. But there are a range of other standards covering sterilisation. There's this 11,000 series covering the sterilisers themselves. I don't propose we cover that today, but we are particularly interested in reprocessing of medical devices, and probably the most complex of the medical devices are endoscopes, simply because they're heat labile. That is, you can't put them in an autoclave, you will destroy them. They're becoming more complex. There was a bit of a move over the last 5 years to see if we could develop single-use endoscopes, but the reality is you sacrifice so much of the functionality that the cost becomes really quite prohibitive. And so I think everyone's settling back down to, we're going to have to come to terms with these reuse issues. And of course, one of the things that turned up in the last 20 years, well, there were let me reframe that. There were 4 groups of infectious or potentially transmissible infectious diseases of concern. The first group really was HIV, which of course goes back to the 80s and 90s. There was a fear that those viruses could be transmitted on iatrogenic or devices of some sort. There was also concerns about hepatitis B group virus. We had some seminal publications in that area, because if you go back to the 70s, the primary way of testing for hepatitis B group was using live chimpanzees. That's not very popular these days. And one of the seminal papers we were involved with through the late emeritus Professor Yvonne Cossart was to develop a brand new method, which is eventually developed into a cell line, a liver cell line out of a particular duck species that allows us to grow hepatitis B group viruses without doing animal testing. But that was a particular concern because those viruses, both HIV and hepatitis B, would be buried in blood. So blood became an issue. The third virus of concern, or really not a virus, but a subviral particle, the transmissible encephalitic subviral particles, things like Creutzfeldt-Jakob disease or mad cow, they also became an issue, particularly after there was a mass transmission in the United Kingdom. And there were concerns about the volume of material that could be transmitted via devices. And there were instances where those transmittable particles were transmitted between patients, both via transplants, so corneal transplant, and also via devices, as well as implantable devices. So that became a bit of a concern. So there was a movement towards making sure we understood how much microscopic material was left after you'd done a reprocessing of a medical device. And of course, the fourth and most recent group has been a lot of emotion around COVID. Those viruses are respiratory viruses. We're really not concerned as much about transmission in this area, but they're still of relevance. What came through all of those things was very defined levels of how much protein you would normally expect as a maximum amount on a device. And let me say, for those that can do the math on this, those volumes are very small and you can't see them. If you could see the organic material, you are way above those levels we've got on the screen. And there's what's called an action level. So anything less than that, 3 micrograms per centimetre squared, is basically no trouble at all or acceptable. If you've got above that 3 and less than a 6.4, well, you need to revise what you're doing. If you've got more than 6.4, it's a fail and you'll probably need to do a recall on those devices. And then under the standards, particularly 15883, there's also a second analyte system you can use. You can do carbohydrate. You can also do ATP testing. So there's an array of things. None of this has really been taken up fully through 5369 in a practical sense, but it's sitting in the background and it's part of the risk profile. And of course, we're really just talking about reusable medical devices, not single-use devices. And the reason why we're talking about those is obviously all of these things get contaminated by biological material. But any contact with skin or mucosal membranes, even with supposedly sterile tissue, represents a risk. And let me say at this point, the most common failure point in a risk sense for a breakdown in these procedures are people, and infection control generally, are people assuming that they are clean and not contaminated in some way. In fact, we're all walking vestibules of microorganisms. And the moment you assume you're safe and clean without going through an aseptic process such as hand hygiene or wearing gloves or barrier protection, you are going to risk failure significantly. And of course, when you're reprocessing these medical devices, they must be reprocessed after every use, and that is before every reuse. Now, in the case of an endoscope, which is that picture there, obviously you're reusing the endoscopes multiple times in a day. These are unwrapped devices. There's a separate expectation for wrapped devices, and we'll get into the Spaulding classification scheme in a minute. The point to make here is that all of these chemistries are reactive, and there are some safety issues. We're intending these disinfectants and, let me say, with the cleaning materials to remove the biological risks, and with the disinfectants to kill the remaining biological risks. And all of these chemistries should be regarded as hazardous. But when it comes to how you process what particular device, this is the global standard. It comes from a paper that was not even peer reviewed actually by Professor Spaulding from about 1967, where he classified the instruments, reusable medical devices, into three groups. At the top end is critical medical devices. So they're the things that are going to come into contact with sterile tissues. So you're going to make an incision and get into a sterile body cavity or area. And for those devices, their standard of expectation is sterilisation, usually by moist heat, not always applicable, but there are alternatives to moist heat sterilisation available. We don't propose to go through all of them today. The next level is semi-critical devices, and that's where most of the commonly reused medical devices out away from the big public hospitals fall into. And they're things like endoscopes, or in the dental field, if you're not doing a dental surgical procedure, you're just doing an oral hygiene inspection of the mouth, most of the devices you're going to use are going to be classed as semi-critical, although in the dental context, most of them can also be heat sterilised. And then there's non-critical devices. There are plenty of those around. There are things like blood pressure cuffs, stethoscopes, and anything that comes into contact with intact skin. And those devices need to go through a lower level of disinfection, but they still need to be cleaned and disinfected. And here's the basic process. It always starts with cleaning. The number one cause of failure for sterilisation or disinfection is failure to clean. If you don't clean correctly, you elevate the risk of failure by literally orders of magnitude. And that's because so many of the bugs that we're worried about will be alive inside biological soils. We won't have time today to fully deal with things like what soils are which and which pose the most risk, because in fact you should look at all of them as proposing a risk, and they should all be removed to the best possible level. Disinfection, as I say, is mainly for heat sensitive reusable medical devices, but there may be other contexts. We're not talking about surfaces here, we're talking about devices. And of course, sterilisation is for things that can go through a sterilisation process, particularly heat sterilisation. And then the fourth part, which is equally important to 5369, is storage and handling, because if you're going through a sterilisation process and you're going through autoclaving, that is a heat and moisture sterilisation, you're probably using wrapping, and those wraps have got a whole pile of other material. Again, we can't cover everything today, but they're going to be wrapped, and there will be limits on how those devices can be stored. For unwrapped devices, the sterilisation or disinfection needs to be done as close as possible to the point of clinical use, so that they can be immediately used and then reprocessed. Now, obviously, there's a whole pile of buying processes here. And let me say, before we get into the detail, from a very technical view, there's not a lot of difference between the old 4815 for office-based practices and 4187 and 5369. The differences are a little bit nuanced in terms of the overarching requirements. The overarching requirements are basically the same: clean thoroughly, disinfect or sterilise, and store and use appropriately. There's a process. And therefore you've got to purchase your right cleaning materials, purchase the right sterilisation system, whether that's standard heat and moisture or a class B, or liquid sterilisers, liquid sterilants, high-level disinfectants. There's an array of things to pick from. The key point to make here, and this is where a lot of people get into trouble, is that all of these high-end products will be included on the Australian Register of Therapeutic Goods. That is, they will be TGA registered. If you're using materials for cleaning or high-level disinfection or sterilisation that are not on the Australian Register of Therapeutic Goods, you're already in the danger zone. And obviously it's up to the registration bodies for professionals as to how they handle that. For non-medical professionals, so things like tattooists and beauty parlours, there's an array of different professional supervision through usually environmental health officers as to what happens there. There's a little bit of flux because some of the EHO bodies are not quite aware of what's going on with 5369 yet, but that's something that'll come up later. And of course, the cleaning agent must be suitable for the task you're choosing them for. So just going out and getting a dishwashing liquid probably does not cut it if you're wanting to clean reusable medical devices. And of course, likewise, the chemical disinfectant, high-level disinfectant or sterilant must be on the ARTG. It must be an instrument disinfectant if you're going to use it on reusable medical devices. You cannot use a hospital-grade disinfectant on reusable medical devices. And in fact, under the TGA guidelines, hospital-grade disinfectants should actually have somewhere on their label a very clear statement that they are not to be used as instrument disinfectants. And of course, when you're choosing the appropriate process, there's obviously an array of, let me say an array, a plethora of legal requirements on the providers of these products for people out in the field. So for anyone that's making a therapeutic good, there is a quite wide continuum of guidelines and standards that apply to things like manufacturing, quality assurance, recall procedures, post-market surveillance. And in fact, even to get something onto the Australian Register now, it's been around for a few years, but people are becoming a lot more aware of it with 5369, requirement for manufacturers of medical devices to validate those medical devices under what's called the Essential Principles Checklist. And these are easily found on the Australian TGA website. And what that really means is your directions for use or your instructions for use or your safety data sheets should all be validated under the Essential Principles Checklist. It's a complex procedure. If you're not running as a manufacturer quality system, it's almost impossible to have compliance. If these products are coming in from overseas and they're on another database somewhere, that does not give them automatic entry onto the Australian Register. They must go back through the TGA. So if you're thinking you can bring something in cheaply from somewhere else because it's approved in Europe or the UK or the US or South America, no, sorry, you run the risk of falling foul of these requirements. And I simply mention that to you because, frankly, it is complex and it is sitting in the backdrop, but it's part of the entire overmantle of what's going on with 5369. And as I say, there's all sorts of other bodies out there covering recommendations as to what can be used and what can't. We don't have time to fully get in that, but obviously people like the Australian Dental Industry Association or Australian Dental Association and the Australian Dental Industry Association, Accord Australia, GENCA for the Gastroenterological Nurses Association, to a lesser extent the ACIPC, that is the infection control people, they tend to fall under NHMRC guidelines and all of the 5369 banner. But there are lots and lots of guidelines out there about what to do. Now, in terms of manual cleaning, and this really is in the now moving into the depth of this stuff, there are products out there. We've got products, we're not the only ones. Here's some names of ours. Obviously, we're aware of what goes on in the dental practices, general practices, day surgery, things for reusable medical devices that can go through a sterilisation process, other things for such as endoscopes. Biofilms are particularly relevant on things like endoscopes. A biofilm, for those that are aware of this and those that aren't aware, a biofilm is not like a biological soil. It's a very specific material that bacteria themselves create, and they join onto a surface and create a little home for themselves. And once they've got this biofilm, they are up to 200 times more resistant to cleaning and more resistant to disinfection and more resistant to sterilisation. Again, we don't have time to get into the full detail there, but we've been involved in this area with quite a few seminal papers in the field, we've been able to show that, for example, in dry surface biofilms, a bacteria that would normally be killed by very low levels of chlorine, say 1 milligram per litre, when you put it in a dry surface biofilm, it will resist 20,000 milligrams per litre of chlorine, available chlorine. So it's 20,000 times more resistant. So obviously cleaning those endoscopes becomes really important, particularly when you've got patients who may have gut infections or commensals that may pose a risk to others, such as transmissible multi-drug infection, VRE and the like. And of course, there's also enzymes. And we do encourage you that if you've got questions, you should go back to the suppliers and ask the questions. Look for the technical data sheets, look for the safety data sheets, ask questions, because frankly, people are trained to deal with this. There's a body of research sitting behind most of the things we're talking about, and we can show you these things. The other big thing that's been a bit of a worry, but not if you've been complying to 4815, segregation of clean dirty. Now, when that standard 4815 first came out, which was, really 2003, there was a big hullabaloo, particularly in the dental sector, about, I've got this little room and how do I get segregation? And so there were charts put up as to how you can have a segregated area even though you're in the same room. Obviously, there were some differences. We moved along. Obviously, in the big hospitals, there are clean rooms, dirty rooms, and there are pass-throughs. Even with automatic endoscopic reprocessors, there are now what's called pass-through devices in the big hospitals. And there was a thought, and it was certainly put out there by the big hospital people, that 4185 was a lesser standard. That was never my view, nor the view of HE023 as a whole. They were just different, and they were aimed at very different contexts of applications. But as the continuum of time has moved forward, there's been basically a lifting of standards everywhere, and a chance, particularly with computerization, to bring all these things together. One of the big arguments in the early days of 4187 and then 4815 was the amount of literally paperwork. And of course, as we've moved forward now, everything's moved to electrical computer-based systems. Some of those paperwork systems have become a little bit passé and have been somewhat obsoleted by modern technologies. And so that started to becoming introduced in 4187, but it had immediate implications also for 4815, particularly on things like traceability and what happens in a recall. And of course, there's the Australian Commission of Safety and Quality in Healthcare. They wanted to have an application, give people the chance to update themselves, particularly around water testing. Water testing became quite a moot point. In the big hospital context, there was quite a move away from filtered tap water through to reverse osmosis systems. That's still somewhat in flux. There's been a whole pile of design issues with reverse osmosis. People think that water systems are pretty easy to handle, but anyone involved in dentistry in particular knows that's not the case. If you don't get your systems right, you can get basically waterborne bugs that are not particularly pathogenic, but will form biofilms in lines and then will host potentially pathogenic organisms. And so there's a whole issue around water testing. And for that reason and others, SQH pushed back the application date. And now we've got 5369. So let's look at what the big issue is. Well, you've just got to get to a single workflow system, dirty to clean. You can't have a single sink that's got clean and dirty in it. You can't do the washing up in the one sink, empty it, and then do the rinsing in the same sink. Yeah, it's just not going to happen. There are some cost implications for that, and that has major implications, obviously, for small practices. But the committee, despite the arguments of people on the committee, such as the ADA representatives and the ADIA representatives and the RACGP representatives, these things have moved to a 5369. However, we need to take note of things like the word should in the standard. Should is not mandatory, and that's incredibly important to make a point of. We're going to come to risk towards the end, but risk is part of that process. There really needs to be a recognition of what risk looks like and how you do risk. And we'll come to that. And of course, water testing is another big area. Now, there were under the 4187 weekly requirements, which are incredibly onerous. And they were onerous for hospitals, let alone for office-based practices. You could never have thought to have got compliance. But the recognition that we've moved to a single standard means some of those issues have backed off somewhat. There still is a need to do some testing, particularly for water, but the frequency has changed quite dramatically, even for the big hospital systems. There are some tests you can do pretty easily yourself, things like water hardness and the amount of chlorine, for example. If you've got a chlorinated water supply, you can get test kits at a very low cost, and this is not complex to do. Some of the microbiology needs to be done fairly regularly, but things like endotoxin testing is backed off quite substantially. And really, the water we're particularly for reprocessing medical devices we're worried about is the final rinse water. So you've cleaned the devices, you've got rid of the biological material. Well, the last thing you want is to rinse off all of the dirty stuff with water that in fact is contaminated robustly with particularly gram-negative bacteria that's eking out endotoxin everywhere. Now that's in reality not common, but it does happen. And it happens where you've got poor water quality in remote locations or badly designed water systems. And you only find out if they're badly designed. Obviously, you know if you're remote, and you obviously know if there's contaminated water. But if the water system is badly designed, you only find that out with testing, and that's the purpose. And of course, you need to do that on new systems when they're installed, and obviously there are different requirements for manual cleaning versus, say, putting in a washer disinfector. And again, this is the two standards, or the two tables out of 5369 that set out the sort of testing required. The table 7.2 on the left, actually most of those things don't require external laboratory support. Most of them you can do pretty easily, but there are more and more people who are offering that service. To a limited extent, we can offer some of that, but it's pretty limited. But it's when you come to doing some of the micro, particularly total viable count, you need to look at how you're going to do that and how that's going to get done. And in terms of frequently, here's really the key chart, how often you need to do it. For WDs, that is for washer disinfectors, well, most of the big clinics will have those, but most GP or dental practices won't have them. And that requires some monthly testing. Likewise, if you're covered by 15883 Part 4, there is obviously some other requirements. And then if you've got an ultrasonic device for doing your cleaning, again, it's about, doing testing regularly. But if you have a look there, it's only water hardness and chloride that you need to test for. And that's got to do with corrosivity of the water. You don't want to leave water on devices that's got a lot of salt, because you will get pitting and corrosion. Now, when this standard, the new one, was published in December, it obviously superseded all of the old standards in Australia. If there are any Kiwis dialing in at this stage, New Zealand has not taken up 5369. So it's just an Australian standard. There is a move, as I understand it, to bring New Zealand in. Not sure quite the timing on that. So the first publication is basically an Australian standard, but it hasn't really picked up. And the goal is to bring everybody under the same basic guidelines. But again, remember, when you get into the standard itself, you need to distinguish between the shall and the should. Shall is mandatory, should is you get to make a decision. here are the broad steps of the standard. It's obviously got a scope, some quality assurance stuff, going through what is the reprocessing issues, what detergent, what disinfectant, what level of sterilisation, getting equipment done, defining everything, defining your processes, validation, which we'll come back to, monitoring, and then how you're releasing products. A lot of this sounds very complicated. Let me say, when you work through the flow of it, it's actually not that complicated. And most places that we see are already doing most of it already. It's just some of the nomenclature is going to change. The one thing that hasn't changed and has been an issue from the start, let me say, so going back to 94, has been what, in the technical sense, is called validation. I remember being told clearly in HEO 23 that validation is quite a simple process. It's simply proving that what you're doing is what you intend to do, and you're doing it every time. And I thought, wow, that doesn't sound complex. But when you get into the technical detail, you find there are at least 3 steps. So if you've got a new device, there's a thing called installation qualification. To my simplistic way of looking at it, that's just turning the damn thing on and making sure that it works, particularly with an automated process. Then there's operational qualification. That's getting a technician probably to make sure that it actually does work and running a couple of cycles on it. And this applies whether you're using an automatic endoscopic reprocessor, a washer disinfector, or a sterilising device of some sort. And then you get to this thing called performance qualification. Now, most of the time, IQ and OQ will be done by the technicians that install these devices, but PQ is probably the most important factor, because that's where someone has to take your standard approach and test that it's working with your standard approach. And when I say that, every clinic will have some differences. Their water quality might be different. The training of the staff might be different. The nature of the cleaning process prior might be different. The nature of the clinical procedures being undertaken may be different, or may be varied. So if you're running a GP clinic and you're doing regularly some fairly simple procedures, maybe some surgical stuff, you'll be running a fairly straightforward set of reusable medical devices. But if you're doing dental work and you're a standard general dentist, you'll have an array of tools that you'll be using, including the handpiece. But if you're doing endodontics, you're probably using a different set of tools. As soon as you get into any of the specialties, well, you know, this load might be your general dental equipment and this load might be your endodontics. So you're mixing them together, or you're keeping them separate. Well, this dentist does it this way and this dentist does it another way. You have to take your load and test it. And there are quite specific requirements as to how that should be done. And it's very important to get that done. Again, we don't have time to go through all of it right now, but if you don't do it right, again, you can have cleaning failure, or sterilisation failure. And again, validation, which is this process, has got these steps, and then it's got documented results. And under 5369, that's been tightened up. And as I say, performance qualification is really performed in your clinic with your equipment and loads, and making sure effectively that you are doing what you intend to do, and you're doing it each time. That is, you're intending to clean and disinfect, or clean and sterilise the devices so they are thoroughly clean and then truly sterilised or high-level disinfected prior to next reuse. Now, there is a whole pile of guidance in this new standard as well. There's some appendixes which set out all the meaning. One of the subtleties about a standard like 5369 is if you look at the front half of the standard, it'll say should or shall, or should or shall. But when you get into the appendix, it threshes out a lot more of the detail around what you might consider needs to be done. And it's quite informative. And you need to read the whole standard together, not just the first part. And as that third dot point notes, it's quite easy to follow because it's set out in the same basic format, so it's not too bad. Now, the big thing that's been introduced and this is applying both to the public healthcare system as well as office-based practices, is a risk-based approach. This is a really significant step change. And let me say, there's been a lot of hue and cry about this in some quarters. But as a manufacturer, I've got to tell you, this is something that's coming through the entire supply chain for therapeutic goods. All of the regulators are moving towards risk-based systems, both proactively and prospectively looking forward, so predictive risk systems, as well as when something goes wrong, doing formal root cause analysis and other risk review systems, whether that's HACCP, whatever you're going to do, you need to have a risk-based approach. And it sounds complicated. And look, it can be as simple or complicated as you want it to be in reality. But let me say, for clinicians, if there are clinicians listening, you already do risk. When a patient comes in to see you, if you're a general practitioner doctor, and they walk into and they sit down at the chair, and you're already looking at the patient for triggers. What's their colour? Are they coughing? What's their speech? How are they dressed? What's their tone of voice? And you're already grading your own response based on what you're seeing. Well, that's risk assessment. And the same goes in dentistry as well. You're already, the moment someone's sitting in the chair, you're starting to do risk the moment they open their mouth. What's the general condition of their teeth? Are they their own teeth? Have they got dentures? Are they properly fitting? Are they poorly fitting? How long since they were last reviewed? So risk is actually something we're doing. It's just that we're now taking these risk procedures and applying them into the cleaning and disinfecting protocol. And again, there's guidance going out there from various of the societies. I know the ADA is starting to send out guidance on how to run risk processes and risk protocols and how to document them. And once you get into the swing of it, it's not that complex, but it is an approach that you need to formalise. And a typical example of that is actually endotoxin. There's a risk-based approach to that. The levels of endotoxin are quite low. There's meters that we can use to measure it. There are cut-off levels, and endoscopes are, not really counted because you're not getting into intact or sterile tissue. And so how you're running your risk will depend on what you're actually doing. So for dental procedures, if you were doing semi-critical and not doing surgical procedure, endotoxin probably is a below risk. And here's the final slide for us all before we move to questions. This is important. It's got a mandatory compliance date of 31/12/2024. That is the Commission, that is the Australian Commission on Safety and Quality in Healthcare, have indicated they expect health service organisations across the country, big and small, to actually have brought themselves into compliance with the standard. Again, there's a should process. Now, for larger organisations who are registered in a particular way, obviously they will need to seek extensions, and there's some outlines as to what sort of extensions can be requested and the reason. But you've got to have your paperwork together. You've got to have your gap analysis. What is it that you're complying with? What is it you're not complying with? So, for example, if your RO system you'd install simply can't get you the water standard, it's going to be a large capital cost. You would go back and say, we can't comply with this part because of this, but we're doing this other thing so that we do have nominal compliance until we can get our RO system up to date. That's expected, but most organisations seem to be moving towards this compliance date. Now, of course, when the new year comes in 1st of January 2025, what will be the outcome? Where will people be? Time will tell. The challenge for all of us in this area is to get everybody up to date and up to scratch as soon as we possibly can. Now, at that stage, I'm going to stop what I'm saying because I'm sure that for those that are still awake, you're sick of my voice and you would like to ask questions. And so I'm going to stop sharing the screen and hand back to our conciliator, Matt, who's going to take over and I think direct the questions. Matt, is that right?
Well, I can do, yes, for sure. Yeah, we don't actually have any questions in from anyone at the moment. Anyone want to ask anything? You must have been very thorough.
I doubt that. I'm too shallow as part of my fear. And look, for those that are still listening, there's a lot of detail in this standard that in a webinar like an hour, we just don't have time to get into. There is, serious science between all sorts of components in this standard. Let me say the standard is very well evidenced. You can't put all the evidence into the standard. It also gets very complex quickly if you want to have all the interlocutory standards. From a purely commercial point of view, we're not responsible for what it costs. We think it's outrageous how much it costs, but that's up to Standards Australia. They're an independent organisation and they set the benchmark for pricing. So that's the way that goes. All right, we've got good question. If ADA is sending an info on risk assessments, who in the GP world is sending it out? Well, at this stage, I don't know of anyone, is the simple answer, unfortunately. I don't know how GP world will cope with that. I am aware that for many GPs, they have started to move to single-use surgical packs and doing less and less in clinic. Now, as soon as you get out of the big cities, that becomes a bit of a problem because you obviously don't have the level of support and infrastructural interconnectivity out in the regions. So you're going to have to do more. In terms of how you run that risk, look, I don't have all the answers, but I think it's something where for those that are in the field, they've got to go back to their regulators and their societies, and whether it's the Royal Australian College of General Practitioners, the Royal Australian College of Surgeons, the Royal Australian College of Physicians, whoever it is, I think they're the people you need to direct those questions to. And if they want to, as honourable societies, liaise with the Australian Dental Association, I know the ADA would be only too happy to have that liaison. So,
yeah, the common question, do GP clinics need a disinfector as well as a steriliser?
Look, I think that's a risk-based question, really. Depends how many procedures you're doing. And the risk is not the risk of failure, because hopefully you're doing your manual cleaning pretty thoroughly as well. The risk question there is, what's the risk in an efficiency sense? If you're doing, 5 or 6 procedures a day, then you're probably going to generate quite a lot of devices that need to be reprocessed. If you're doing 5 or 6 a month, it's hard possibly to justify the economics. And I think that's where you need to contact people like Southside Surgical, who are going to be able to help you happily with those sort of questions, and others. In terms of service agents, obviously your suppliers know who they are. That's okay. For facilities that don't have a designated clean and dirty area, you better start looking at capital cost, is my recommendation to you. You might need to think about a redesign. Now, that's going to have a capital cost implication. And again, if you were seeking exemption, that's the sort of thing you would seek exemption on and say, look, we're in a particular size. We need to redesign. We need some help. It's going to take us 6 months, but, it's going to cost us so much money. And, in the meantime, we're doing a couple of things. And again, seek advice from the sales staff who are going to support you with product selection, because some of those people are actually incredibly skilled and well trained. I know our people certainly are, and they'll be able to give you quite significant technical advice on how to use products correctly and overcome some of those difficulties while you're sorting things out. Is distilling your own water? I think that's a great question for sonication and sterilizer. Okay, sure, as long as you validated it. It depends how you're doing your distillation. I mean, distillation is a very old way of dealing with it, but you've got to store the water correctly as well. So, and the problem for distilling the water is it doesn't take out all the ions. Now, if you're going to do deionization, which I don't think anyone is going to do unless they're a manufacturer, then you're probably okay. And this is the big thing between different systems. How do you take out the bugs, but also take out the salts and other residues, particularly in regional locations where the water quality can vary substantially, even between time of the year. If you're in Mount Isa, for example, and it's bloody hot and dry for a lot of the year, and then a cyclone mounts through, your water quality is going to change quite substantially, pretty damn quickly. And of course, the clean water will last. The volume of fresh water will last a long time. But if on the other hand you're running off bore water, you need to deal with those issues. Next question: if you've done 4187, do you need to redo it? Well, you've got to have compliance to 5369. So the simple answer is yes, you have to do it, but you have to do it against 5369. So the first thing to do is what's called a gap analysis. That is, you look at what you're doing 4187 and look at 5369 and follow it through and find where you're complying and find where you're not complying. Now, if you're complying on everything, that's fantastic. Well done. Hats off. But you also need to document that. So you'll need to say, I comply with this section, I comply with that section. And if you're really thorough, you might say, I'm complying by doing this, I'm complying by doing that. But you'll need that sort of documentation. I'll come back to why you need the documentation in a minute. If you've found things that you're not complying to, well, then that becomes a gap. And you then start to look into how you're going to comply. And part of that's then looking, it's a risk analysis really, is say, well, how am I going to get to compliance? So yes, you need to have done, I would have thought, at least a serious gap analysis prior to the end of the year. Ultrasonics are not mandatory. They're just recommended. Frankly, they're a fantastic cleaning device if you've got instruments that are going to respond to them. But there are some issues to worry about. Things like dental burrs clean fantastically, but you've got to be very careful about needle stick injuries, so we don't recommend that. But I know a lot of dentists are still doing that. You've just got to be careful of how you're doing it. Reprocessing non-sterile items. Well, I think let me reframe that question from LW, from Lynn. I think you need to put it in the Spaulding classification scheme, Lynn. So are the instruments we're talking about going to contact sterile tissue? Are they critical? That's a yes or no. Are they going to contact intact mucosal surfaces? That's a yes or no. If they're going to contact only intact skin, then no, they're not sterile. But you still need to reprocess them. And yes, they should be cleaned and they should be disinfected. There's lots of literature on, for example, stethoscopes harboring multi-drug resistant organisms, Staph aureus of commensal. I mean, there's literally hundreds upon hundreds of papers in the literature on stethoscopes and what they will carry with them. Part of that's because people don't look dirty; they just look like people. But as I said during the talk, we're all walking microorganism cabinets. And we've done and published research that shows when you touch a dry surface biofilm, let's say with Staph aureus, when you touch that biofilm, those bugs will stay on your fingers for up to 19 subsequent touches. And to make matters more complex, we've also published data that shows that in a typical ICU, 90% of the surfaces that are commonly touched will have a biofilm, a dry surface biofilm, that has an MRO or one or more embedded in the biofilm. So they stick to your fingers and they're out there. And it's the same with devices that contact skin. These bugs are on skin and they will get onto the fomites. Wonderful paper published by Andy Lee et al. In the journal Infectious Diseases and Hospital Epidemiology from Australia, in an ICU, showed the transmission or cross-transmission of vancomycin-resistant enterococci, largely via two things: hands and reusable medical devices that weren't being reprocessed. So the fact that they're Not critical or not high level does not mean they don't need to be reprocessed. Quite the contrary. They should all be reprocessed, and they should be reprocessed regularly. Will the compliance be tested through accreditation or a different method? It'll be largely done by accreditation. There's no compliance authority out there independently, and Standards Australia aren't a compliance authority. They simply publish standards that are commonly agreed standard by all the stakeholders in the field being involved. And the case of HEO23, for those that don't know, if you look up the number of people on that standard, it's a huge committee, huge. Dozens upon dozens of people sat on that committee. Dozens of organisations have been involved over a huge number of decades. And so, but they all come from accreditation bodies, and depending on who you are will depend on who your accreditation body is. If you're a daycare or centre, you might have the Australian Department of Health, you might have New South Wales Department of Health, or Queensland Department of Health, or Victorian Department of Health, but it will be either a state or a federal body. If you're an individual clinician, then you'll be covered under the Australian clinical group now. There's a new clinical arrangement. It used to be done with all the individual groups, people like the Dental Board of Australia and the subsidiary under that, and under each of the state bodies. That all now goes back up to a national body. But the people who come out there are normally either federal or state people who have enforcement powers, and they're the ones who will be doing the inspections. And of course, they do these things routinely and randomly, but they also respond to criticisms or complaints. So if you get a complaint and it's seen to be substantial, you might have a visit from one of the regulators, or from the notifiable body, or from your registration body. And it'll vary by clinical area or specialty. Next question. Can you confirm that the vital validation process needs to be done by the steriliser technician and not by the practice nurse? That's a should, not a shall, that one. So there are all sorts of process technicians out there. Some of them will do this well, but most of them will do it, and most of them will charge a fee. So when they do it, they will charge a fee, but you should get a report that becomes part of your documentation. If you're going to do it yourself, particularly if you're doing steriliser loads in an autoclave, whether that's a Class B or whatever, then you better understand what your process challenge device is. You better understand how you determine what your hot points and your cold points in the chamber are, and what sort of process control device you're going to use, whether it's going to be various classes, or if it's a Class B, a Helix or a Bowie Dick. Some of this stuff can get very technically complex. So my personal view is that if I was in clinical practice, I would find the best technician I could find and get them to do it for me and get paper out of them. And if I had to pay for it, that's a safer outcome for me. That's my experience and observation, but that may not be everybody's view. I think we're just about out of time, aren't we, Matt?
Oh, we've got one more question there. If you can answer that in a minute or two.
Yep. We use stainless steel tubs for dirty instruments. Do we need to put them through the steriliser, or is it clean? Well, again, it depends on whether they're critical, semi-critical, or non-critical devices. Now, if they're non-critical, they don't need to be sterilised, but they need to be at least subject to some sort of disinfection, and with an instrument disinfectant. Now, there are a range of devices out there where they use low-level instrument disinfectants. I know those products are out there. We certainly have some. There are others that do as well, or you may use a high-level disinfectant. A good example of that is reusable face masks in a range of activities. For example, in mines rescue areas, they reuse their masks, so they go through a process of decontamination and disinfection. And that's because the masks are used between different people, so you don't necessarily have your own dedicated mask. If you've got tubs and you're doing the instruments cleaning and your dirty tub is here and then you rinse them off in the clean tub, I'd like you to just tell me you're doing it with running water rather than dirty water, because the first time you do the rinse with in the clean tub, the whole tub's contaminated on the basis that it homogenizes. So you want to be testing the last instrument, not the first instrument, rinse cleaned. But should they be subject to a decontamination and disinfection process? Yes, and they should be done regularly.
Awesome. Well, we're right on an hour now, so thank you very much, Dr. Whiteley, for that. That was a lot of information, and it's great to tap into your knowledge and experience, and hopefully everyone's gained a lot from that. So keep an eye on your inbox, everyone. We'll send out some information next week. Yeah, and we can all.
And Matt, thank you to Southside Surgical for hosting such a wonderful opportunity seminar.
Not a problem, not a problem. All right. Thank you, everyone. Have a good day. Bye-bye.